Maybe for these people who are joining us for the first time and they don't know who we are the German Human Genome-Phenome Archive. And our mission is to provide and to maintain a platform for human omics data sharing and everything in compliance with the GDPR. We aim to make clinical omics data open for the research, and in doing that, we hope we can facilitate the secondary use of this data and contribute to biological discovery and replication of these findings to show the validity and translation of research in science in genomic medicine. And of course, we can do it everything alone. There are a lot of and diverse communities involved in that, involved in helping us to set up this huge infrastructure. And for example, some of these communities are computational biologists, molecular geneticists, clinicians, scientists, physicians, and of course, the patients. And that's the reason we are setting up this seminar and also other events. We have next month more events. For example, we have events on site. We will have our symposium here in Heidelberg on 15 October. And it's free for registration. You can join us in Marcelius College. We have a lot of great speakers. And we also have events virtually, so you can also join us, join our lecture series. Next month, we will have as a speaker Anthony Brookens from the University of Leicester, and he will introduce some information about research assets discovery in large European networks. So if you are interested and you want to join us, you can do it via our website, ghga.de/events, or you can also sign up to our newsletters and keep up to date with our events and follow up. You can do it also via our website or just scan this QR here. And now I would like to introduce our speakers today. Our first speaker is Karoline Mauer. She studied her bachelor in biology at the University of Bonn and also her master in organism biology, evolution, and paleobiology, also in the University of Bonn. She's currently a PhD student at the Deutsche Zentrum von neurodegenerativen Erkrankungen, I think. And she's since July 21 a member of GHGA and is working in the metadata work stream. And our second speaker is Anandi. Anandi Japan. She got her macrobiology, her bachelor in macrobiology at the University of Madras, and also make a master in bioinformatics, and also in life science informatics at the University of Bonn. She had made her PhD in applied life science by Fraunhofer. And she's also since May 21 member of GHGA and metadata work stream. And this is a pleasure for me it's a pleasure for me having you today. And I hand over to you. Thank you very much.
Yeah, thanks for the introduction. I will start today. And the webinar is split into parts like we already heard. So I will start with the first part about fair biomedical data. And because the topics are a bit not really disconnected, but a bit different from each other, we have a first Q&A session after my talk, and then Anani will take over with the fair data portals, and then we also have a Q&A session for her, and also there's a feedback poll, I have been told. Okay, so I will start with the FAIR biomedical data and just a bit of a structure. So I will start with data and metadata and then go into a bit more detail about biomedical metadata. Then I will cover the FAIR principles, why they are important and how you can make your data fair. And in the end, since we are the metadata workstream, I will talk a bit about metadata models, what solutions already exist, and if you really feel the need to create your own, how you can do that and what you have to consider. Okay, starting with data and metadata. In a very general definition, data is only potential information. It is raw and unorganized and mostly only a combination of characters and symbols. Since we have really no clue what kind of data we are looking at, we also don't really know what we are supposed to do with it. But once you add context to data, it becomes information. It's now processed and human consumable, and information can be measured, analyzed, and visualized for specific purposes. You can also try to answer different questions using information, and you can make it useful for your purpose. And once you ask how information can be useful for you, you leap from information to knowledge. Knowledge is information that can be applied, for example, by asking how the data and information connects to other pieces of information that are already out there, or how you can use the data to achieve your goals. And once you use the knowledge and make decisions based on it, it becomes wisdom. To reach wisdom, we have to answer questions as why should we do something, or what is the best course of action, again, to reach my goal. And on top of that, data and information are like a look back into the past, whereas knowledge and wisdom are more of an outlook into the future. And in order to walk up this hierarchy from data to wisdom, it is of course not enough to only have the data, but you also need context and extract the information with processing and contextualizing. And for this, you need the description of the data, which is data about the data or metadata. Yeah, it's a bit abstract, so I brought a short example. Let's say you have an RNA-seq experiment, then your data, the raw information are the FASTQ files. You know that there's potential information somewhere in there, but right now, as I said, it's just a sequence of symbols, and you first have to extract the information that is in the FASTQ files. So to get the first step, the information, you would already need some basic information, such as the organism you're working with, or also the ensemble version you're using to annotate your genes. And using this metadata, you can get from the raw FASTQ files to normalized and annotated gene counts. And you can also plot your first graphics for quality control, for example. And the second step within this information is then to do an analysis, maybe a differentially expressed gene analysis. And for this, you would also need to know the patient groups or the disease you're looking at. Then the knowledge domain in this example would be the classical discussion part of a paper. So you try to relate your findings to whatever is already out there in the literature. And you can also examine where your results close gaps or answer questions. And at this point, at the knowledge domain, you are not only reliant on what you found, but also other people's information and data. And finally, the wisdom part would be to use your knowledge and build the future based on it. Ultimately, you might want to use your data to develop possible treatments based on the genes you identified. And for this, you would need to know the limitations of your treatment, of your study. You would mind to conduct follow-up studies or replication studies before you go into the treatment development. And from this example, I hope you can see that you need different kinds of metadata for different kinds of contextualizing while getting from data to wisdom. So this brings me to biomedical metadata. And looking at metadata from a researcher's view, we can roughly divide it into different kinds based on the data that is being captured. We have data about the individual, which might be demographics or clinical information. Then we have technical data, such as the data type, the instrument models or pipelines that were being used during data generation or analysis. And then we also have the administrative metadata, which tells us why, where, and what data was collected, and then also who's the responsible person for it. And it's hard to define core fields for it because biomedicine is a very vast field, but over our work in GHGA, I think we've come to more or less the conclusion that if we were to define core fields for the individual or the sample, these would be age, sex, disease, case control, and sample tissue. For the technical data, you would need to know the data type, instrument models, pipeline, software and software versions. And for the administrative data, you would need to know the study type, the data controller, place of collection, the kinds of data modalities that are in there. So different kinds of RNAs, or different kinds of sequencing, different kinds of clinical metadata, demographics, and so on. And you also need an abstract that ties everything together and explains it in a bit more detail. And having these core fields makes or helps to make data fair. So why do we need to be fair? For those that don't know, FAIR stands for findable, accessible, interoperable, and reusable. And it basically means that others can find your data, they know how they can access your data is readable for humans and machines and can be interacted with, and your data is well annotated and can be used for diverse use cases. The FAIR principles were first published in 2016, and important to note here is that they were meant to act as a guideline for those wishing to enhance the reusability of their data, and they are aimed at the machine's ability to use and find data, and thereby then aiding humans. The FAIR principles were developed and also needed because we are experiencing an explosion of data. We still are. And we have increased international collaboration, which both lead to data management issues. And another thing to note, just for the completeness sake, is if we talk about FAIR data, we always talk about FAIR digital objects. And one digital object consists of 4 components. If we keep the example with the RNA-seq experiment, then the data would be the sequencing matrices, then we have the metadata and the provenance. Provenance are, for example, protocols, and we have a persistent identifier. And in this FAIR context, one digital object is only fair if all of these 4 components are fair. And we all know that this is difficult in biomedical sciences. So why is fair still important, even if it's difficult? In other words, why would you even want to share data if it's so difficult and almost impossible to do so? First of all, I'd say it's part of the scientific process. To validate your findings, it is important to know where your data come from and how you process them. And also it's important for others to have the chance to validate what you found. In turn, others might be able to close gaps in their knowledge with data or with data that you shared. Another important point is that you can lead by example. By sharing your data, you can encourage others to do the same and then in turn benefit from the increased data availability. And ultimately, fair data advances science and it also builds trust, which is often overlooked but also very important. What happens when data is not shared can be seen in the reproducibility crisis. And here I took a paper from the reproducibility project Cancer Biology. In principle, they took 193 experiments from high-ranking cancer papers that were published between 2010 and 2012, and they tried to replicate them. And from these 193 experiments, they were only able to replicate 50. And the various reasons for that you can see on the right-hand side. What they found is that among the studies that provided sufficient information to be redone, the effect sizes were on average 85% smaller than in the original findings, and the original positive results were half as likely to replicate successfully than original null results. Here you can see what I found very striking is that all protocols needed clarifications, and in more than 50%, the data was not shared. And you can only speculate why the data was not shared, but I found another article by the editor-in-chief from Molecular Brain, and he wrote about his experience with data submission. He wrote that over his time as editor-in-chief, he requested additional raw data for 41 manuscripts. And of these, 21 manuscripts were immediately withdrawn without providing any raw data. An additional 19 were later on rejected because the raw data was not sufficient or it had mismatches with the results that were reported in the manuscript. And his conclusion is that there's a possibility that in some cases, the raw data did not even exist in the first place. So talk about trust. But even if the raw data existed, just getting them online somewhere does not automatically make them reusable. Because if we do not have the appropriate infrastructure and policies to organize the metadata, then most data will remain not usable because it's simply not findable. So this is where we come to the part where I tell you how you can make your data fair. And I want to preface this by saying the most obvious thing first. It's that not everything can be done by you if you're a PhD student or a researcher. You can, of course, make your data interoperable and reusable because this depends on the standards that you can implement. But making them findable and accessible depends on the infrastructure, and also it depends on the kind of data you're working with. So to make data findable, you first have to clarify whether it's even allowed to be findable. For example, if you have detailed clinical data or social data, then it might get difficult. The findability, as I said, also depends on the solutions that are implemented by a data repository. So do they have a proper search engine? Are they proper indexing? Do they have keywords? Do they perform vocabulary wrangling? So can you search with synonyms and still get the same results? And to make it accessible, again, you have to clarify whether it's even allowed to be accessible or which restrictions apply, and if yes, what kind of restrictions these are. And the technical accessibility depends on the solutions that are implemented by the data repository again. So do you have open protocols? And what does the metadata lifecycle look like? So can you access metadata even though the underlying raw data might have been deleted? To make your data interoperable, it's important to standardize your vocabulary. You can either do that by using ontologies or you define your own controlled vocabularies. Then you can follow a data model to describe and structure your data, and you don't even have to develop your own because there are a lot of models for different kinds of data out there. For example, if you want to structure clinical data, you can use OMOP or FHIR, but you can also use phenopackets, which are very flexible for any kind of clinical data. And I would also really recommend to use the data dictionary to explain the data and the metadata, just for you to record what you did and what single columns mean. And to make it reusable, you should make sure that your data is well annotated, you capture detailed metadata as far as it's possible, you use domain relevant community standards. This might also be that you store sequencing data as FASTQs and not as TXT files. And you should link everything to a publication with information about the methods, the results, and the limitations of the data. So the Too Long Didn't Read about things you can do and things you just have to accept is that if you have any leverage on it, might not be the case, but if you have, before you collect your metadata, maybe think about what your data could deliver beyond your own project and then make a metadata wish list. You should definitely harmonize and standardize your vocabulary. You can use a data model if your use case needs it. You should have a data dictionary. You should use the domain relevant standards. You should, of course, publish. And if you want to upload the data later on, choose your data portal wisely. Things you just have to accept is that detailed annotation of your metadata might not be possible. For example, if you have real-world data that comes, for example, out of hospitals where people are super busy and don't have enough time to go over your very detailed metadata catalog, then you might not get the data you want. Anandi also will have a slide about this very shortly to describe the differences between cohort and real-world data. Then you might run into the issue that the standards have not yet been accepted in your research field, or there are still competing standards, and you might just happen to use the wrong one. And accessibility and findability might be limited by GDPR and ethics, depending on the data type you have, or if you have data subjects which are sensitive. If you're interested in these data management plans and how to record any decisions you take, then we have a video about that with the folks from Elixir, but you can also find it in the GHGA YouTube channel. Okay, and this already brings me to the last part about metadata models. A metadata model is a representation of data, and it allows us to structure data, define relationships between entities, which are real-world objects, so a person or a protocol or a sample. And you can describe these real-world objects using properties. For example, for the individual, properties would be sex, age, or diagnosis. And metadata models are needed when you want to store and query data in a database. There are already existing metadata solutions. We have, for example, agnostic solutions. If you think back about the fair digital object I had in one of the first slides, then you can use OwlCrate. So it's not a metadata model per se, but it's a way to generate these digital objects by packing your data and your metadata and your protocols and everything else together in one package. Then the other schemas.org, LinkML, JSON-LD, and RDF allow you to build your own metadata schema, but one word of warning is that they might be too comprehensive if you just have like a little table. Then I would advise against using that because they are really extensive. If you have provenance data, so you work a lot with pipelines or workflows, then you can check out the workflow OwlCrate or biocompute objects. And if you have biomedical data, there's ISATab, which also has a nice section to record any experimental metadata. There's bioschemas.org. OMOP Fire and Pheno packets I already mentioned, but you can also check out the Kerndatensatz by the Medizininformatik-Initiative or the NFDI for Health data catalog. If you still feel the need to develop your own metadata model, I have a short how-to for you. So first is you should identify your data. So if you are dealing with a certain disease or are working with a certain type of data, then you can already check out other data portals to see how they do it, and this will already give you a nice idea of where you should go. Then you should identify the necessary compatibilities. Again, if you always upload to the same data portal, it might be useful to just do what they do, adapt their structure, and maybe add one or two entities you really need. And if you already use one representation over the other, then don't really go for something else. Pretty self-explanatory. You should identify already existing standards. So if there are any community standards that already exist for your experiment or study type, you can adapt those. For example, if you're working with sequencing data, you can check out the minimum information about any sequence standard, or MinSeq, or when you use single cell data, you can check out MinSCE. Then identify your entities. So which real-world objects will you capture information about? And then after you have your entities, you can think about the properties. So which information you need to describe your entities. Then when you have your properties, you can standardize your vocabulary, because nothing is worse than having a metadata model with only free text. I recommend the ontology lookup service to find suitable ontologies, but you can also use it to define your own vocabularies. Or if you have your own LIMS system, you can also check out what they have for their vocabularies. And lastly, if you have to link your entities, do it as a last step. This is really required when you want to store and retrieve data, and you should find a representation that works for you and allows you to extract all the information you need. There are some pitfalls and things to consider. First, I mean, I cannot stress it enough, but aligning to standards and other schemas is the most important thing. Make use of everything that is already out there. And I'm not saying that you should never develop something new, but if you have to, your use case might not fit into what is already out there. So yeah, if your use case doesn't fit and you really have to develop something new, make sure that it is still aligned to whatever was already developed. Second, ontologies and controlled vocabularies are necessary. So why develop a metadata model if you're not going to control what people put in there? Then third, remember that especially in the biomedical field, accessible data doesn't mean that it has to be open. To be FAIR compliant, you have to be accessible. Your data doesn't have to be openly shared. And just because data is open doesn't mean it's accessible, because it can still be in a format that no one can open. And lastly, remember that human data is considered sensitive under the GDPR. It requires special protection and processing contracts. Again, it depends on your use case and what you want to do with it, with your data. But if you have any questions or feel unsure about something, know who your data protection officer is, get in contact with them and discuss with them. Yeah, and if you have any further interest in FAIR or data management plans, we have additional resources on the GHGA YouTube channel or on the homepage. So yeah, feel free to check them out. And this is it for my part.